We have got 45 folks here, and we're expecting more, but I think we should start. And I just want to welcome everyone. And I want to introduce a few people who are here. I've mentioned Don Lockheim, who's mayor of the village of Sagg Pond, Ann Welker, who's on our Southampton Town Trustees, Jackie Fenlon, who works with for the town planning department, and also is I think coordinating the CPF water quality improvement program. We have staff here, Matt Swain, Josh Halsey, Amanda Abraham, and there may be more coming, I'm not quite sure. Bruce Horwith, who's our consultant working on the Sagaponic restoration work. I see Kim Cordy, who's on our staff. She's joining us as well. For those of you who do not know the Trust well, we were founded in 1983, and we've protected over 13,000 acres on Long Island. We are often confused with the Community Preservation Fund. So I'm here to tell you that the Trust does not receive the 2% real estate transfer tax. All those funds go to the town for conservation purposes and water quality purposes. Our work is supported primarily by private contributions. We work certainly very closely with the town. Trust has a long history of conservation work on Sagg Pond. With the assistance of 9 neighbors, the Trust acquired Smith Corner in 1989 from members of the Foster family. And I believe Lee Foster is here. Lee has been involved with the Trust since its inception, and the Lockheims as well. They've very early on in the early days of the Trust. So we own 2 preserves on the pond and hold conservation easements on about 5 parcels in close proximity to the pond. And we are really privileged to be playing a coordination role toward the revitalization of Sagg Pond, thanks to funding from the Joyce and Irving Goldman Family Foundation. And I think Kutch is here, and Ben Swanger. A big thanks to you for really underwriting our work to pull this all together. And there are other donors as well, the Drexlers, the Myers, the Baldwins, the Gouges, Barbara Phillips, and more, who have supported the Trust's work in this effort. Working with the Stony Brook Foundation, 19 of your neighbors have raised $224,000 to launch Chris Gobler's work, his research of Sagg Pond. And I just want to acknowledge all of those folks who well, I'm not going to name them all, but the fact is that they have really enabled this work to start over the last 2 years. And so you should be proud of them. There's going to be a lot more work to be done in the coming months and years to get Sagg Pond back to its pristine state, if we can ever get it there again. We're certainly going to work hard. We're also going to be working closely with the town. The town has committed $112,000 towards Chris's work, and you know, it's very gratifying. But one of the reasons why we're here tonight is really to educate you about the critical role that you play as residents in the watershed. And we really look forward to hearing Chris's preliminary findings of his work. And there's a lot more to do, so we're going to keep you engaged. And with that, it's my pleasure to introduce you to Chris Gobler. And Chris, you want to maybe introduce yourself for folks and then take it away.
John or Chris, if I could just break in for 1 second, a couple of housekeeping items. If you could turn off your mics while Chris is giving his presentation, that would be great, just so we cut out the background noise. Please feel free to use the chat feature. I will be monitoring that and can flag questions for Chris. And then at the end of the presentation, we'll ask that you all turn off your mics and we'll have a conversation and you can ask questions that way as well. So thank you. Go for it, Chris.
Okay, very good. Well, thank you so much for the introduction, and good evening, everybody. It's nice to see everybody, some new faces, and some old friends as well. And excited to give you this presentation about what we've been up to in Sagg Pond. We've really been working on Sagg Pond for several years. The presentation today is really going to focus largely on our findings for mainly 2019 and 2020. I'll start by acknowledging all the people who are working in my laboratory group at Stony Brook University, a whole array of graduate students, postdoctoral scholars, research technicians, who are the boots on the ground and the fins in the water, and really make all of this happen. And you're welcome to follow along on all the things that we do in social media if you're interested in some of the topics we're going to talk about today. So I'm going to begin with the overall concept of a watershed. And the idea here is that any water that falls within the land surrounding any water body is going to ultimately find its way into that water body. And so that's sort of shown here in this diagram. And so what you can, the overall take-home message that, the rainfall, be it on your yard or your neighbor's yard or people upstream from you or downstream from you, all of that water has only two fates, or maybe three, but they're all critically important. One, that water is almost 99% going to end up in your groundwater. So that's the water underneath our feet. And that is your drinking water. And even if you get public water, that public water is not coming from many miles away. There's no such thing as city water. People call it that, but you'll see, I'll even show you where your water is either coming right from your backyard or from wells right in your community. And I'll show you where those are later in the presentation. And the water that we don't use for drinking water, as you can see here, ultimately discharges into coastal waters and discharges into Sagg Pond, for example. And what's happening around the town of Southampton and all over Suffolk County are the following two trends. The population obviously is growing. And as the population grows, the amount of nitrogen that's in the groundwater continues to increase. And so this is just data for all of Suffolk County, but this same trend can be found on the South Fork and around Sagg Pond. More people bring more nitrogen. And I won't go into all the details, but you should never presuppose where that nitrogen comes from. In fact, we know where it comes from for Sagg Pond. That's part of my presentation this evening. But these high levels of nitrogen we're starting to see are problematic for two reasons. One, high levels of nitrogen can even be a human health concern if it's high levels in drinking water. But as we're going to talk about today, the big issue at hand is the effects that high levels of nitrogen can have in the coastal zone. And so if you look at this map here, this map was made by my laboratory documenting the occurrences of what we call water quality impairments that occurred across Long Island just in 2020. So 2020 has been an unusual year, as we all know, for many reasons. Frankly, this map is not that unusual, however. This is all things that occurred between June and September of this year. And this isn't me making a judgment call, but rather this is the occurrence of something known as a harmful algal bloom. So the growth of algae to a level that can have a negative effect on the ecosystem, or low oxygen conditions. And either way, be it the harmful algal bloom or the low oxygen, it can be traced back to high levels of nitrogen. And again, I'm not making a judgment call here. I'm using state and federal standards, making measurements and comparing it to that. And so I just bring this as context because you can see Sagg Pond is on here. And you can see it's in good company with other areas that are also having low oxygen conditions and harmful algal blooms. So the overarching goal of the project that I've been engaged in is to get as much information as possible to come up with a plan to both manage and restore Sagg Pond. Frankly, we come to the table with there being very little data about the pond. And so if we see something that we would call an impairment, deciding on how to fix that impairment would really be guesswork without data. So the whole purpose of this project is to get baseline data, and also with regards to particular impairments, find out what the causes are. And so breaking this down, we looked at water quality within the pond, focusing on bacterial levels, algae levels, and oxygen levels. We performed experimental work to try to understand what is controlling the growth of algae. We undertook an approach called microbial source tracking to figure out where the bacteria in the pond are coming from. We did something similar with regards to nitrogen and phosphorus. Today, I'm just going to focus on nitrogen, however, figuring out where the nitrogen going into Sagg Pond is coming from. And then finally, we did some work on filter feeding bivalves, specifically oysters. And as I'll talk about later, they can be an important component of water quality restoration. And they have been used as such in many cases across Long Island. So on the first front, looking at the water quality conditions within the pond, we're using an actually fairly innovative approach. When we began first sampling Sagg Pond, before this project began, I would send a graduate student there. They'd collect a water sample, stick a probe in the water, get some information, maybe come back a week later or 2 weeks later. And that would give us a decent record. But for this particular project, because of the investment that was made, we were able to construct and install a real-time water quality monitoring buoy, solar powered, that makes measurements every 10 minutes from the moment we put it in the water until the moment we take it out. And the data that it records is not just recording on the internal device, but it's then sent to a website. So my laboratory has a website. I will say for now, however, being in the off season, we've ceased monitoring the pond. We don't want to see the buoy get caught in the ice, and with the impending storm and winter weather, it's out for the season. But in season, the data goes to our website, and within that website, you can actually select Sagaponic Pond, make decisions, excuse me, about looking at all sorts of water quality parameters, including things like temperature, salinity, dissolved oxygen, pH, nitrate levels, levels of total algae, levels of blue-green algae, etc. All really critical because things change very quickly. As I'll show you momentarily, if you're just making a measurement once a week or even once a day, you're not getting the full picture. So for example, let me show you the levels of salinity in Sagg Pond from this summer and fall. Now, as you can all intuit, that salinity measures the amount of salt in the water, and a freshwater body like a lake or pond would be 0, and the coastal ocean around us is around 30, 31. And as many of you, or hopefully even all of you would know, Sagg Pond is temporarily open to the ocean several times a year. The Southampton Town Trustees open up the cut to the ocean, letting ocean water in. And so what you can see here is that for this data set in particular, through the summer, the levels of salinity were quite low, not 0, but as you can see, just above 5 parts per thousand. And then a sudden increase in the middle of August. Well, we can all guess what that was. We don't need to guess, I can tell you. That was when the cut was open. And you can see at its peak, the south end of the pond where the buoy was or is kept reached nearly full ocean salinity. However, that cut did not last very long. And as you can see, it was opened in mid-August, and only a week or so later that cut closed. And you can see very rapidly the system responds, begins to fill up again with fresh water. So you can see it's less than a month, just a matter of 3 weeks by the time the salinity gets down again to, in this case, around 10 parts per thousand. It was opened again, but then closed again shortly thereafter. So these openings, they're wild, so to speak. Sometimes they can last for a long period of time. Other times they close shortly thereafter. But these openings can have a very strong effect on the conditions within the pond. And so moving on here to dissolved oxygen, if you look at this data, it may simply look like squiggles, and you may say, wow, that's a mess, that's bad data. But no, actually what this is the natural range of oxygen in Sagg Pond on a day-night cycle. And the reason this is because during the day, when the sun is shining, the ecosystem is experiencing lots of photosynthesis, and that drives the oxygen levels up. And then at night, in the absence of photosynthesis, the oxygen levels drop very, very low. And for perspective, you can see the readings here in milligrams per liter. But for perspective, I'm putting in here what the New York State Department of Environmental Conservation tells you the level that the oxygen level should never go below. They recommend the oxygen level should never get below 3 milligrams per liter to maximize the health of aquatic life. And frankly, they also say ideally levels would stay above 4.8 milligrams per liter. But using that lower threshold, what you can see is that you can see an overall trend with levels decreasing. And just before the pond was opened, we had what I would call nocturnal anoxia. So that's at night, the oxygen level is going down literally to zero. And that's a dangerous situation for any sort of aquatic life. But you can see also how the pond responds to the opening of the cut. The levels shot up and were very significantly higher. They began to decrease again when the cut opened, they increased again. And then the other thing about oxygen is that typically in the absence of biological activity, oxygen levels are inversely proportional to temperature, meaning that in the summer, they're low anyway. And so that's why I'm really focused on summer here. You'll never see low oxygen conditions in winter because the natural background levels as the temperatures drop get quite high. And so that's partly what you're also seeing here at the end of the season as the waters cool, the oxygen levels begin to increase. But clearly the existence of these low oxygen conditions, and in some cases, no oxygen conditions, just demonstrates that this system is under stress, likely associated with high nitrogen and high levels of algae. Now, one particular class of algae that we have great concern about in Sagg Pond is something known as blue-green algae or freshwater cyanobacteria. And I'm showing you two of the most notorious of those right now. You can see the scientific names, Microcystis and Anabaena. And the reason we worry about these types of blue-green algae is that they make these bioactive compounds, secondary metabolites, toxins. Specifically, you can see microcystin, which is a gastrointestinal toxin, and then anatoxin A, which is a neurotoxin. Now, when these toxins were first discovered in the 20th century, before they had the official names, scientists first called them fast death factor and very fast death factor, because when they isolated the compounds, the effects on living organisms was quite rapid. And you can see they've since been renamed, but despite the more mild sounding names, they can be serious compounds in communities where they have to rely on surface waters for their drinking water. If that drinking water is contaminated with some of these compounds, it has been linked to cancers and linked to having carcinogenic effects. But even if you're not using it for your drinking water, we also would worry about our pets. The CDC released this study just a few years ago, documenting that there have been dozens and dozens, hundreds of cases of poisonings of dogs due to these blue-green algae. And in fact, if you're not aware, just a few water bodies to the east in Georgia Pond, there was the death of a dog due to blue-green algae in 2012. And it seems nearly every summer we have at least one report of a dog getting sick from drinking water from some water body across Long Island. So this is another reason we worry about these compounds. And beyond dogs, these toxins are potent when they're implicated in the death of very large mammals. So I don't know if anyone followed this news, but over the summer, there are reports coming out of Africa of elephants dying over the plains of Africa in the country, in Botswana. And it was a mystery through the summer. It started in July, the reports continued, it went from dozens to hundreds. And then finally, by the end of the summer, beginning of fall, the scientists there were concluded that the elephants had expired due to the consumption of water tainted with these blue-green algae. So in this country, these temporary ponds form seasonally following the rainy season. As they evaporate, everything in that pond gets more and more concentrated. And you can see here towards the end, as the pond is just about gone, and by the fall they were gone, you can see the condition that the pond is in. So it becomes very shallow and very potent, and unfortunately led to the poisoning of these, unfortunately, actually endangered animals. So within Sagg Pond in 2020, this is the weekly measurements of blue-green algae. Now, these are weekly as opposed to on the buoy because my lab is charged by the New York State Department of Environmental Conservation of reporting out on blue-green algae for downstate New York. So all of Long Island, but also in New York City, including Central Park and Prospect Park in Brooklyn. And so what you see here is a typical seasonal pattern. These blue-green algae like it hot. So the warmer it gets, the more likely you are to see these events. And so what you can see is we go from almost zero level, a reading of almost zero in the spring, a steady increase going into summer, and then a peak. And you can see the levels peaking above the threshold. So above 25 micrograms per liter, the DEC will issue a warning that that water body has a blue-green algae bloom and should not be recreated in. And you can see here, I'm putting the genera of the blue-green algae, Aphamas amenon, that was found at that time. And note, very importantly, when the cut was opened in mid-August, we went to a period where the blue-green algae were flushed out and the pond was really clear of blue-green algae for 2 weeks. But once the cut was closed and it began to fill up with fresh water once again, we had the resurgence of the blue-green algae. In this case, a second genera known as Anabana dominating. The cut was open again and they went away again. And so I was remiss to not say it yet. I'm just say it now that in addition to these blue-green algae loving the warm temperatures, these are really what we call fresh or brackish water algae. So you won't find them in the ocean. You won't find them at high salinity. They like it at a salinity usually about 15 or lower. And remember, for perspective, the ocean is about 30, and of course, fresh water is 0. So when you open up the pond, you're really creating a condition where they can't grow. But when you close it, particularly during summer, that creates the possibility for these blooms to occur. And just wrapping up just a few other facts about these blue-green algae, beyond the genera that we showed you in past years, we've seen these other 5 genera present within the within Sagaponic Pond. We have measured the toxin microcystin at up to 1 microgram per liter. This thankfully is below the recreational standard for a water body, but is above the drinking water standards. The EPA would tell you this water body is not fit for being a drinking water source. But again, thankfully, nobody was doing that. But clearly, with these sorts of conditions, we want to know what are the causes to therefore figure out what we can do to solve the problem. So to figure that out, we conducted some experiments, showing you here a picture of another regional water body with these blue-green algae. This is Lake Agawam in Southampton. So we're not alone in Sagaponic Pond by any means. And in the literature, what you'll find is that either nitrogen or phosphorus might be important for controlling these events, depending on the water body. And there's a lot of variabilities that can control this, including the types of blue-green algae you would find, as well as the levels of salinity, the latitude you're in. So you really need to, instead of guessing, you really need to figure it out. And so we used a well-established method, an experimental method, where we collect water and then we either alter that water by adding nitrogen or adding phosphorus or adding both compounds. And then essentially, and also looking at 2 different temperatures, because I already mentioned to you, temperature can be important. And then essentially assessing what happens over time. Do you get more or less of the blue-green algae? And so here's just an example from one of the experiments that we conducted this past summer. And so what I'm showing you here is in the different treatments where we added nothing, nitrogen and phosphorus or both compounds, the final levels of biomass that we saw in response to these different elements. And as you can see, when we added nitrogen, it led to a significant increase in the amount of chlorophyll and blue-green algae in the water, suggesting this is the primary limiting element. You can see when we added just phosphorus, almost no effect. A slight increase when we had nitrogen and phosphorus together. So we might consider phosphorus secondarily important to these blue-green algae, but not as important as the nitrogen alone. So knowing how important nitrogen is, the next important step here is to figure out where is it coming from, right? And really, it's so important to get the answer to this question correct. Because if you want to solve the problem, you don't want to be chasing something that has nothing to do with the problem whatsoever. So we made actual measurements and did some modeling to try to understand what's the relative importance of the sediments at the bottom of the pond, wastewater from on-site septic systems, fertilizer from people's lawns. We also looked at fertilizer from farm fields, surface runoff, birds that live on the pond, atmospheric deposition. And we looked at both the atmospheric deposition that's going on land and actually directly in the pond. And interestingly, on the one hand, you might assume atmospheric deposition, that must be when it rains. And that's true, but as it turns out, half of the nitrogen that comes by atmospheric deposition comes on just any old day. So there's a constant rain down of particles that can contain nitrogen that can also be a source to the pond. And we also looked, of course, at the primary tributary at the headwaters as well. And I'll just emphasize also that I've done these sorts of studies for many water bodies. It's been done for many water bodies on Long Island. And within, I would say, a couple of dozen miles from Sagg Pond, I can point to you and point out water bodies where the most important source is wastewater, the most important source is fertilizer, and yet another water body where it's actually atmospheric deposition. So it's important we don't presuppose these things. I'll mention for our sediment survey, we surveyed from the neck of the pond all the way down to the main part of the water body. And what this is showing is that data in GIS format, showing you the depth of the mud within the water body, and with red being the thickest mud. And in good news here, the large part of the main body of Sagg Pond, it has almost no mud. It's mainly sand. And that makes sense because the ocean is probably bringing a lot of sand in there. That's where the most tidal range is experienced. And so it doesn't really allow mud to accumulate. And that's a good thing. We don't want mud because it can be a strong source of nutrients. And this plot shows the amount of organic matter within these sediments. And this is a proxy for the amount of nutrients it may hold. And again, in good news, there's very little organic matter in most of the pond, save for the neck, which is an organic rich area. Now, beyond looking within the pond and the sediments, the next important thing we needed to do was to look at the watershed. And that's what's shown here. Hopefully you can see in the blue outline. So this is the entire area that contributes water to Sagg Pond. And you can see it's really big. It's not the homes. It's not just the homes that are on the pond. And in fact, you can see there are homes that are very far away and field areas that are very far away that are contributing water to Sagg Pond. You'll also see some holes, right? Here's some areas that are not contributing water to Sagg Pond. And you might be surprised to see that, but actually what this represents are what are known as community supply wells. So your drinking water, if you're getting water from Suffolk County Water Authority, they have 2 well fields within the watershed. And these areas, because the well fields are there and they're drawing water to supply drinking water, they do not contribute to Sagg Pond. The water that falls there is simply used for drinking water. But there are these areas that are both close and far away that eventually will find their way into Sagg Pond. We were even able to contribute how long does it take for the groundwater to get to Sagg Pond. And that's what's shown here, and it's color coded. The areas in blue and the lightest purples are all 20 years and less. So it takes a while for the groundwater to get there. But conversely, there are even areas that are north of there that are taking even longer than that, more than 50 years for the groundwater in the very northern extent to make its way all the way to the south. So when we built our model, we had to collect a lot of data within the watershed. So I'm showing you just some of that data here. But we did a lot of work using GIS, Geographic Information Systems, and getting data from the town, from the state, and even doing some ground truthing data, making some measurements ourselves as well. And then we also used a lot of what we call just basic data parameters for the model. And importantly, I am not doing this as a sole entity doing guesswork on all of this. We're in an era, thankfully, where I showed you all of Long Island has water quality impairments. And thankfully also we have governments from the town level to the county level to the state level that is playing a very active role in addressing these issues. So as an example, I've been involved in the Suffolk County and Nassau County, what they call sub-watershed plans, where I've been meeting with scientists from places like Cornell University, the US EPA, the USGS, the Department of Environmental Conservation, the state of New York, people within Suffolk County, people within Nassau County. And we've had committees where we've sat down and we've thought about how is nitrogen affecting all these different water bodies. And all these different parameters you see here, all these different numbers are not my best judgment calls. It's not me making a guess, but it's what we call the scientific consensus that this committee came to, where all these scientific minds are coming together, looking at the scientific literature, both locally and globally, and figuring out what are the best numbers to use for modeling within Long Island, within Suffolk County, within the town of Southampton, and here for Sagg Pond. And I could talk about this for a long period of time, but suffice to say, we are considering essentially every possible fate for an atom of nitrogen that might fall within the watershed. So with all that, here is the punchline to where's the nitrogen coming from. And it's a really interesting pie chart here. It's not quite like Trivial Pursuit, but it actually is looking close to it. You can see, because there's a balance here. Our estimates are that agriculture is actually the largest source of nitrogen, but right behind it are on-site septic tanks and cesspools. So those are the two biggest sources, and probably, even though it's 29 versus 23%, within the error range, you can say they're almost equivalent. But we also see that lawns are an important source, the atmosphere is an important source, the sediments are an important source also. But clearly the biggest ones are both agriculture within the watershed and the on-site septic systems. And as I mentioned, every system's different. I believe if you were to go, for example, showed Lake Agawam before, the answer there is it's mostly septic systems. Wainscott Pond, the first water body to the east of Sagg Pond, it's agriculture. So it's different no matter where you go. And supplementing these, where the nitrogen's coming from, we also got some data to map out the concentrations of nitrogen within the watershed. And with this sort of map, you can see some hot spots just to the northwest of the pond, as well as some elevated regions in the very furthest reaches of the watershed. And typically, when we see these sorts of hot spots, these are often associated with agriculture. It doesn't mean that's the only source, however, in the end, it's really about this pie chart here. And again, agriculture and on-site septic systems, which every single home has. I should just mention, because I won't get to it later, but a quick word about these septic systems. Everyone has their own septic system, and these were designed, literally, this is early 20th century, the systems that we have is early 20th century technology. They're meant specifically to make sure that the wastewater from your home gets away from the surface waters and down into the groundwater as quickly as possible. And that was designed for protecting human health. So that's a good thing. But it also is literally injecting that wastewater into our groundwater, which, as a reminder, becomes our drinking water and then discharges into surface waters. And you can see its effect is significant. And for all of Suffolk County, the biggest source of nitrogen is on-site, and Nassau County, at least in North Shore Nassau County, the biggest source of nitrogen to surface waters is these on-site septic systems. And again, you can see some hot spots that are likely related to agriculture, but some of these also certainly being related to on-site systems. I'm going to move on and just give a quick presentation about oysters, because as I mentioned, it may seem like a hard right turn here, but as we think about remedial approaches, filter feeding bivalves can be an important approach here. And again, I'm talking a lot about regional water bodies. I've talked about Georgia Pond, Sagg Pond, and I'll now mention Mecox Bay, the water body just to the west of Sagg Pond. And what Mecox Bay has in common with Sagg Pond, of course, in addition to being very close, it's one of Mecox and Sagg Pond are the two water bodies that are temporarily open to the ocean in the town of Southampton. And the third one like that on all of Long Island is Georgia Capon in East Hampton. I bring up Mecox Bay here because Mecox Bay has the most productive oyster population from a fisheries point of view of any bay in all of Long Island. There's active commercial harvest of oysters in Mecox Bay, and this has been the case for many years. And it's partly because of the lower salinity in Mecox Bay. We talked a little bit earlier about salinity. The ocean's about 30. What we're learning is that oysters are prone to disease, and the diseases that infect the oysters thrive at higher salinity. And therefore, when you have a water body like a Mecox Bay that's temporarily closed to the ocean and fills up with fresh water, it keeps that salinity down and it allows the oysters to thrive. Also keeps away predators. But the important thing about these oysters, besides the fact that maybe many of us enjoy eating them, they're also what we call ecosystem engineers. They are filter feeding bivalves. They filter the water. That's how they feed. One oyster can filter 250 liters of water in one day. And you can see the image down here. This is an experiment that I conducted many years ago. It's actually a 300-liter tank. You can see with just a handful of oysters in the bottom of one. We had none in the other one, and you can see how those oysters can keep, hopefully you can see the water line here. This is filled with water, but that water is crystal clear because of the filtration strength of these oysters. Now, we only began the work with oysters this summer, so we don't have our full data set yet, but I'll just give a quick overview of some of the things that we found. We had oysters at two different locations, one in what I call the center of the pond by the bridge, and another in a more southern site. We had them out this summer looking at their growth rate, their survival rate, the presence of disease. I believe we still have one of the cages still out there. We had some problems with the cage in the middle of the pond. People were very curious. We had some people come out and multiple times pull the oysters out. And as aquatic organisms, they tend to not like that, although they can live for at least some time out of water. We also had a surprising event. I told you how dangerous blue-green algae can be to humans, to dogs, to elephants. It looks like the oysters did not like the blue-green algae either. We did have a significant dieback of some of the oysters during one of the peaks of those blue-green algae blooms. So we learned a lot this year. We're still working on the data, and we're going to be putting these out again to learn more, because despite some of the bumps in the road this year with regards to the tampering and the blue-green algae, the early data on the disease presence in Sagg Pond looks very promising. So again, following what we're learning about all these systems is that these lower salinity systems, the oysters are a lot less prone to disease. Okay, the last type of data I'm going to talk about here is on bacteria. So I talked about blue-green algae. They're also sometimes called cyanobacteria. These are what we call heterotrophic bacteria, and specifically the New York State Department of Environmental Conservation measures what are known as fecal coliform bacteria. They're known as indicator bacteria, potentially pathogenic bacteria. That allows the DEC to make declarations whether a water body should be kept open or closed for shellfishing. You can see Sagg Pond is in the uncertified category. So there is no shellfishing allowed in Sagg Pond. Again, likely because of bacteria levels. So one of the questions were, well, what do the data levels actually look like, and how does it compare to standards? So here's an example of one of those classes of bacteria, here known as Enterococcus. We have here the New York State Department of Health guideline for swimming. And so you can see, and this is the 2019 data set, but that we had levels that were at and sometimes above that threshold through 2019. And when we looked at the another kind of bacteria, these fecal coliform bacteria, we compared that to the DEC standard. And again, we saw the levels were higher than is permitted for shellfishing. So sort of affirming the DEC's declaration of not having the water body open. We also looked across the pond to look for trends and potentially identify sources that way. So here are a couple of, I should mention, so cruises with 5 different stations from the north to the south. And so did that twice last year. And you can see in the first cruise, maybe an expected pattern, high levels at the north and then declining thereafter. But on another date, we saw following a rainfall event, pretty high levels across the board. And then we looked at yet another class, the Enterococcus, the one that's used for swimming. Somewhat similar patterns, but a little bit divergent as well. But certainly getting those measurements that are above the levels permitted for swimming. So it's good to know that these affirmed the prior discoveries. But really, what we want to do is, again, the whole purpose of the study isn't to collect data for just the sake of data collection, but to get the data that can inform action that can be taken to improve water quality. So the state agencies only tell you, yeah, there's high levels of these indicator bacteria or not, but it doesn't tell you where they're coming from. So we used an advanced, what we call molecular genomic approach here to not only look for these bacteria, but use, I'm sure everyone's heard of the idea of the microbiome. Look at the microbiome of different types of animals and look at the bacteria that are resident in different types of animals, and then come up with a fingerprint for bacteria that are from deer, from birds, from humans, and from dogs, although I'll say that the dogs share a microbiome or have a microbiome that's similar to small mammals. And our molecular probes, DNA-based probes, can't differentiate across those, but essentially looking at which class of these animals is contributing to those high levels of bacteria. And here is the punchline. So here's a data set from last summer, going from May all the way through the fall. And here are the different classes. And what you see mainly here is gray. And the gray is the marker for the dogs and small mammals. And again, small mammals would include mice, rats, raccoons, etc. And so we see that the very large majority of those fecal bacteria are coming from the dogs and small mammals. We also see an interesting trend. If you squint your eyes, you can perhaps see that in the beginning of the season, it's almost exclusively those organisms, whereas later in the season, we do see a little bit of a human contribution and a bigger contribution from birds. And so what this tells us is that those, those septic systems which are delivering a lot of nitrogen are actually not delivering too much in the way of bacteria. And we're also seeing, there's a pattern we've seen in some other water bodies on the South Fork of Long Island, that late summer into fall, you actually get birds migrating through and have a higher population of birds in the region. And they seem to be contributing to some of these bacteria. And when we did 2 different north to south cruises from the across the pond, I showed you these before, and compare the summer and the fall, you can see particularly in the south, more of the bird signal later in the fall. So that's the data. And so I'll give you first some conclusions here. I've showed you that there are blue-green algae blooms in Sagg Pond. We are seeing the compound microcystin, which is a public health concern. Some of the chlorophyll levels are higher than we'd like to see. The oxygen levels are getting lower than we'd like to see. And that the algae in the pond are being promoted primarily by nitrogen, maybe secondarily by phosphorus. The biggest sources of nitrogen to Sagg Pond are both agriculture and on-site septic systems. We'll be updating that model as we go, and that's the beauty of having the model done. Now with the model done, we can both correct it if we get better information. For example, we'll work with the Peconic Land Trust to get more information on agriculture, and we can also now play out scenarios. What if we upgraded septic systems? What if we cut fertilizer? We can then see what the effect might be on the water body. I showed you there are elevated levels of fecal bacteria in the water body and that they're coming mainly from small mammals and secondarily from birds. And then we did see some plumes of high nitrogen in the groundwater as well. From here, I'll just mention a few next steps. At the university, in addition to being a professor within the School of Marine and Atmospheric Sciences, I'm also the director of the New York State Center for Clean Water Technology. And one of the most, our primary function for the center is to develop technologies that'll do a better job of removing pollutants from water streams before they enter our source water. So before it gets in our drinking water, before it gets into surface waters. And one of the biggest things that we focus on are on-site septic systems. And we're focused both on developing systems that do a better job of removing nitrogen, but also we're recently moving into working with communities to try to get as many homes upgraded to systems that do not leach high levels of nitrogen into the groundwater. And I think there's going to be an opportunity for doing that in Sagg Pond. A couple other things to consider that we've worked on, in addition to continuing the bivalve work, there may be opportunities to be moving seaweeds and other sorts of algae out of the pond as a source of nitrogen and phosphorus. We can also be looking at sources of surface runoff that are contributing those bacteria into the pond. And then finally, thinking about best practices for fertilizer use and ensuring that both for homes and for farms, the fertilizer that's being used is the right kind and contributing the minimal amount of excessive nitrogen and phosphorus to groundwater. So with that, I'll conclude. And if there are questions, I see the chat is fairly full here, not full, but there's questions in there. Be happy to take those questions. So
Yeah, Chris, maybe I'll start off with asking the first one that came in there. If that works.
okay. Yep, great.
Thank you so much. That was really, it's the third time I have listened to that, and it's always very interesting. I always learn something new. Nancy Bynum, you talked about the toxicity of blue-green algae to dogs. Have you had any reports of people having reactions, and what are those? What about wildlife like birds, fish, and turtles?
Yeah. So to my knowledge, there has not been any negative human exposures. The most common route to being affected by this blue-green algae is oral. So, and so the times where dogs have gotten ill, and I probably should have clarified this, but now I have the opportunity to do it. It's not only that the dogs are drinking the water, but these blue-green algae have this tendency to float to the surface of the water. And then on top of that, the second thing that can happen is if you have a persistent wind, and in summer, we often will get winds off of the ocean blowing north that can concentrate the algae on a shoreline. And so the situations where a dog might get ill is that not only is the blue-green algae there, it's concentrated on the shoreline, and then the dog may go in and either, it could almost be a solid or a foam. The dog might eat that and or be licking that up. So it's that oral exposure that really gets to them. And for reasons that scientists have not figured out yet, dogs sometimes will tend to, they see this and they start licking it up, whereas seemingly the indigenous animals know to avoid it.
Interesting. Are there any follow-up questions to that? If not, I'll move on to the next. Okay, so Barbara Bornstein, to everyone, are you considering the underground movement of water within the chain of ponds in the Long Island Pond Greenbelt? And why is septic usually targeted while agriculture contamination is hardly mentioned in the community? I could read those both separately if you need me to, Chris.
Well, I will say on the second point, it really is that the septics are, in many cases, are the biggest source. But again, that was the whole purpose of this study, right? It makes no sense to presuppose. And had we not done this study, we would have been on the front of only thinking about the septics. But we know that that's not the only source, and we need to be, in this case, thinking about agriculture and addressing that in parallel. And we are, when it comes to the different ponds that are there, I believe that some, the ones that are in there are in our model. So we are considering those. So any of the water bodies, the ponds that are in the watershed are part of our model and were considered, and anything outside of that boundary was not considered.
Okay, so from Roger Baxter. So with residential septics and pets, I assume we are talking about urine as the primary source of nitrogen. What's going on under the hood of agriculture? So again, two questions in there.
Yeah, so that's right. I mean, just to get into the weeds, When it comes to waste, yep, the urine has about 80% of the nitrogen. And so it really is, that's where it's coming from, whether it be humans and or pets. Under the hood of the agricultural fertilizer, I mean, it's just the amount that's being added. I will say for the model we use, we got really particular. So we're not, that is to say, we're considering the types of farms that are present in any given plot. And we have for different types of crops, the standard nitrogen loading that's used for those types of crops. But no matter, even though many of these farms are actually, they become, I will say, I'm not trying to make them out to be the villains. Many of these farmers have gotten very sophisticated over time. Look, it's in their best interest to not over fertilize as well, because they pay for that fertilizer. They don't want it washing out. But you can't be 100% efficient with the fertilizer. And so, so the fertilizer that makes its way to the pond is essentially the nitrogen that is not utilized by the crops and is washed through the ground. In almost every case, it's not really runoff, but rather getting washed down into the groundwater.
Okay. Thank you. So from Christina Mala, this is fascinating and there's a lot to think about. Would Dr. Gobler consider giving a talk outside on site at some point in the spring or summer? Also, I see people crabbing at Sagg Pond. Any thoughts on food safety for people who are crabbing at Sagg Pond?
So on the first point, yeah, giving a presentation outside to lots of people sounds like something I would really embrace. It's been a year of Zoom talk, so I'll look forward to seeing everybody in person, hopefully in 2021.
Perfect. It's a date.
Yeah, yeah. You name the place, I'll be there.
Cool.
And as far as the as far as crabbing goes, this is it's closed to shell fishing for filter feeding bivalves. I don't think it's closed to crabbing. And in most cases, it's there's no regulatory reason to close it unless there's a blue-green algae bloom. And I think this has come up before, the idea that we need to make sure that everybody knows when those events happen in summer. And I know that we have got Ann Welker here with us, and the trustees give, in addition to the health department, the trustees also give guidance on use of the pond. So I think it'll be I think we identified that last time. We need to make sure that when there are one of these events, the proper signage is going up so everybody knows.
Right. Barbara Bornstein, since Canada geese stay here year round, would migration still be a factor in late season bird contamination?
Yeah, probably not for the in residence Canadian geese, but there are other birds that migrate through and are here temporarily. And apparently, like I said, I'm not a bird expert, but I have spoken to some people who do study these things. And apparently, our particular part, or that particular part of the South Fork, is a thoroughfare for birds migrating beyond just Canadian geese.
From Deborah Aller, when you match up the different nitrogen sources into Sagg Pond with the groundwater travel times in the watershed, what percentage of the nitrogen input do you think is legacy nitrogen? And how might you expect these different proportions of nitrogen to change over time as we have seen more development and less ag in the region?
Yeah, that's a great question, very astute question. And so we really built the model on really 2020, 2019 loading rates. And therefore the model doesn't account for legacy farming, for example, because we can't manage the only thing we can do is manage for the pond we have today. But there may well be that in the past, the nitrogen loading rates in the past could have been higher or lower. If there was very intensive agriculture, it could have been higher. But I think as Debbie was alluding to with her question, that it's also possible that, in some cases, more homes could be contributing more nitrogen as well. But again, our model was not looking, it wasn't looking retrospectively, but rather was really focused on what's happening today.
Okay. From Barbara again, you mentioned fertilizer. What about pesticides and herbicides used on lawns and in particular farmlands?
Yeah, so I mean that's certainly, you know, there's no doubt that that's happening. But really the models we built were strictly focused on nutrients. So you know, not something we're really accounting for. I will say in the Center for Clean Water Technology, we have uh, a whole initiative looking at drinking water and looking at, you know, those sorts of what we call organic contaminants or potential organic contaminants of drinking water. So that's, you know, something we could look at in the future, and it may well be that the some of that data is available through Suffolk County as well. That we relied on Suffolk County Department of Health Services to generate the map that we had, and there may be other contaminant data to draw on from that.
Okay. And Barbara did write also, fantastic presentation. Thank you. We have a question from Mark McIntyre. To summarize your presentation, what causes the trustees to open and close Sagg Pond and Mecox Bay to the ocean?
Well, again, I think Ann Welker could probably speak to that better than I. I have worked with the trustees for 20 years, and I therefore know that they definitely, firstly, want to make sure people's homes aren't flooded. So, I mean, people who live right on the water, they may have had the experience where if the water levels get very high, there could be water in people's basements, for example. So that's certainly motivation. I know in Mecox Bay, they also look at the salinity, and they try to keep the salinity around or not much lower than 10 for the benefit of oysters. Although I've had discussions with the trustees recently that, you know, if it gets to 5, it's probably still okay there. But I know those are really usually the two of the main driving factors. I also know that during the summer, you know, if the water level is consistently low, there's just less ability for people who live on the pond to be able to recreate in the pond. So that is, I know particularly for Mecox Bay, when the cut is open, people who live on the creeks who had water in their backyard one day may have a mud flat the next day, and that's not great for boating. So I know, at least for Mecox, that's also a consideration. I don't know if Ann Welker, who's with us, wants to offer anything further on those points.
The one other thing, I mean, what restricts us that's tricky about opening the cut is the endangered species season from April 1st until September 1st. In a perfect world, we would open more during that time, but we are restricted to opening. Harmful algal blooms, Chris, speaking to Sagg Pond, we keep our eye on those, and when we see them, because we know that when the cut is open to the ocean, that the temperature and the so since when the salinity and the temperature drive those harmful algal blooms, and when the cut's open, we know that that makes a tremendous difference. So I would say feel free to reach out to me if you wish to discuss that further, Mark.
And I know somebody had a question earlier today about how do we ensure the cut gets made regularly and early enough to prevent flooding, but I believe that answers that question pretty well, and thank you. Another question that came to me was, is there a way to install a permanent dam or other structure to control water levels in the pond? Leave that out to Ann or Chris.
It's a good question. Permanent dam to control water levels. I don't think that would work, actually, because I think the pond is filling in not just from you know, the headwaters, but there's also just groundwater inflow. So I don't, you know, you wouldn't, you're not going to stop the water, I guess is the bottom line there. So I don't think that would be enormously effective.
So you'll hear a dog possibly barking in the background, so I apologize, but such is life on Zoom. Stefan asks, great presentation, thanks. It seems that Sagg Pond is getting shallower. Does it make it hotter and thus more prone to algae blooms? Does it need dredging?
So that's a great question. And yes, it probably is getting shallower. And yes, that makes it hotter. And dredging, that could be an interesting approach. Now, on the one hand, oftentimes I told you we did a sediment survey. We often do that because in some cases, the sediments can be a strong source of nitrogen. And so I may come out and say, if we found that, for example, that was the biggest source of nitrogen or a source of phosphorus, when we were very worried about phosphorus, we might come in and say, yeah, dredging should be a top priority. I don't see it as being a top priority necessarily for combating nutrients, but as a parallel story, I will say in East Hampton, over in Georgia Pond, they recently got their 10-year dredging permit, and beyond opening the cut, they made a very conscious effort to also be dredging out that whole south area, because what happens, of course, is that when the pond is open to the ocean, the ocean waves come in, they're carrying sand, the energy of those waves slows down in the pond, and then that sand settles out. So it's only natural that these sorts of water bodies will accumulate more sand over time and start filling in. So it probably makes sense over a long period of time, eventually to do some dredging, again, maybe in that southern end of the pond.
Okay. All right, Doug Halsey with the big words that I have to try to pronounce. So Chris, help me. Are Cryptosporidium or Giardia significant factors in Sagg Pond's bacterial contamination?
So I am pretty sure that, well, one, we didn't look at those. So I don't know. I will say those are usually drinking water issues and fresh water. So I'm going to guess that they're probably not, because of the levels of salinity that are in there. And I can combat his big words by saying I'm pretty sure those are protists and not bacteria, but that's a little we can discuss later.
From Marilyn Clark, is Mecox Bay less polluted? If so, is that because of the oysters?
So Mecox Bay does not experience the same consistency of blue-green algae blooms. And I think the oysters probably contribute to that. I think other things that contribute is the fact that Mecox Bay is just a bigger water body and a little bit deeper. And so there's this calculus that comes from the kind of the ratio of the watershed to water body volume. And so when you've got a big watershed contributing to a tiny water body, it's going to have a bigger effect than if you have a larger water body and an equivalent sized watershed. There's also the nuance of what's going on in the watershed, but the one thing that Sagg Pond is working against it is that it is quite small and shallow, and therefore more sensitive to runoff and any groundwater flow when it comes to nitrogen and phosphorus.
Okay, Barbara again. Hi. Does dredging also backfire by sterilizing the life in the pond?
No, I would say no. In fact, maybe the opposite, in that this dredging and opening of the cut, it's a little strange, but it's been going on for, they say that when the settlers came, they learned it from the native populations. And what you get by doing that is you actually get an influx of some ocean life into the pond. And oddly, for example, for the blue crabs, we still haven't figured out how this works. But for blue crab populations to survive, their life cycle is supposed to be that the larvae, so the baby blue crabs, the microscopic baby blue crabs, are meant to go out to the ocean as larvae and then develop and then migrate back in as small crabs. So kind of like a salmon, but not really, obviously. How they do that in something like Sagg Pond, not really sure. I think, they're probably, those populations are probably shared between Georgia, Sagg Pond, and Mecox Bay. But anyway, point being that opening of the cut is, because it's nothing that lives in the pond, everything that lives in the pond is used to the salinity fluctuations. So it's not leading to a mass die-off, and instead it probably is maintaining some of the populations that are in there, like the blue crabs, for example.
What about dredging to actually make it deeper as opposed to the regular opening of the cut?
Yeah, and so I think that is something that could be considered. I know that, I don't know. I need to talk to Ann about what their long-term dredging permits from the DEC look like. But like I said, in Georgia Pond, they made an effort to not have their permits not just be to open the cut, but to dig out some of that sand. And the nice thing about that is that actually that is, and my data affirms this, that's nice, rich ocean sugar sand. So that's not, sometimes when you dig up material like mud, you have to figure out where you're going to put it because the DEC won't let you put it anywhere. But because that's sand, it's actually kind of valuable. So there could be, that would be easy to not just dispose of, but it could be used to nourish the beaches. So there could be some opportunities there. And I see Ann nodding and affirming that as a concept. So I don't know if you want to add to that at all, or give a more nuanced, detailed view there.
I don't know as much about this, but I'm learning fast. And I know that when the way the permit is written, there is permission for a 1 year or an annual dredge that's 1,000 feet straight back in the center. So that would take away a lot of the flats that exist there now. I don't know if any of you have been down to Sagg Pond in the past week or so, but with the cut being open right now, there's a huge flat that's exposed. And if this annual dredging were to take place in a perfect world this year, it would help with tidal exchange, and that would benefit the whole ecology of the pond.
So Chris, we still have questions coming through, but I know you've been talking for a while now.
I'm happy to answer more. We're here. That's why, it makes it, yeah, no problem.
Okay. I know you've been on internal Zoom for like 48 hours. So you're like, whatever, I'm fine. All right.
It's fine. I mean, it's a good opportunity to get everybody, up to speed.
Okay, great. So Casey wrote, how much can one balance how these bodies of water act without human interference, i.e. the cut with the need to alter them to human benefit? Is the need for the cut because homes have been built in what would naturally be wetlands and flood areas?
Yeah, that's probably on the mark. What would it probably happen, or probably what happened before any settlement, is that naturally the pond would fill with water. And then at one point it would get so full and then a storm would come, the ocean would breach it, and then you'd have the natural exchange. And then over time, what they call natural sediment transport processes in the ocean would close it. And the whole process would start again. So my sense is that it's probably, obviously there's a human element. The trustees are driving this to make sure that there's no flooding and for other reasons. But the overall concept of it opening and closing is actually quite natural, and that's the case for the entire south shore of Long Island. In fact, even though there's only 4 permanently, or 5 if you count the Rockaway Inlet in Brooklyn, permanently open ocean inlets. It's estimated that there's been dozens and dozens of different inlets in different locations over the last several hundred years. And so it's just this is the way the south shore of Long Island functions, really.
All right, so where am I on these? Let's see, Deborah. Do you think edge of field PRBs strategically placed on the neck of Sagg Pond might be one solution to reducing nitrogen inputs into the water body?
Yeah, potentially. So a PRB is a permeable reactive barrier. And so the concept is that if installed in the right place that has high nitrate in the groundwater that's flowing in a consistent basis, you can essentially dig a trench to intercept that groundwater, fill that trench with wood chips that then lead to the removal of that nitrate via a process called denitrification. I will say, however, the Center for Clean Water Technology has done work with this, both on our own and in collaboration with Cornell. We just graduated a doctoral student on this exact topic for her PhD, Molly Graffam. And what we've learned, it's a lot more complicated than that. And so we need to find out things like how much oxygen is in the water. Is the nitrogen ammonium or nitrate? What are the flow rates? How deep down is the high levels of nitrogen? And I'll just give a quick corollary that we're doing a study in Lake Agawam where we thought, oh yeah, PRB, this is going to be perfect. And we found high nitrate for sure, but it was mostly ammonium. And I won't do a chemistry lesson, but I'll just say that you can't remove ammonium with wood chips. You have to go with a whole nother route. So a barrier probably won't work. So it's a good idea. I'm hopeful that will be part of the solutions here, but we need a little bit more information. And thankfully the Peconic Land Trust has brought on Ron Paulson from Cornell Cooperative Extension, who's an expert in groundwater. He's going to be getting probably the information needed to answer that question.
Yep, and Ron's here. Hi, Ron.
Ron, you want to add anything to what Chris has said?
Yeah, no, Chris, I think did a good job of, laying out the difficulties and challenges. It's not something that's going to work everywhere for all the geochemical and hydrological conditions that you need to do. But I think it does have a place, and it could be very effective. And I think Sagg Pond has a lot of potential for that. And I think, the data, and we work cooperatively with Chris and his folks in the labs and Molly Graffam. So I'm excited about continuing that and getting the data, and, the challenge of it is certainly there, but we've learned an awful lot just in the last 3 or 4 years. So I think, it has potential. I look at it as a triage sometimes to do while we do these inland things that we need to do, IAs and other things. So I'm looking forward to continuing that effort with Chris and his team.
Thanks, Ron. All right, 2 more questions. So Gary Sherman, you mentioned the BGA foam along the Sagg Pond. Is this the same brown foam that can sometimes occur along the ocean shoreline as far away as Gibson Beach?
No, that would probably be different. There's all sorts of marine foam, ocean, bay, freshwater. And I should emphasize that the foam I was describing for Sagg Pond, that was just hypothetical. So I really meant that as a description of what can happen in the cases when there is a dog incident, dogs getting ill. It's often because of the accumulation of foam at the shoreline. I wasn't referring to that specifically happening, although it can happen. But it's going to be different than what you'd see in the ocean, and actually in the ocean, it's actually due to another type of algae. So these should be facts that you know when you study algae, different resulting foams.
All right, so Roger, is a systems approach you have seen elsewhere worth looking at in solving the Sagg Pond problem? i.e., are there factors that can be introduced or reintroduced that might help create a more sustainable, self-correcting ecosystem in the area? Bad example, when endangered wolves were reintroduced to Yellowstone, they helped solve the overpopulation of other species that were causing damage to plant life, etc.
Yeah, I mean that's a great question, and it part of this is partly the philosophy with regards to the oysters, right? And so because I didn't mention, but we're also going to be doing some survey work to understand what are the natural bivalves within this system. And so, maybe there's already a resident oyster population. If there is, it's not very dense. We know that. But the idea, this is part of the idea. Can we rebuild stocks of oysters or other bivalves, maybe mussels, and again, we wouldn't want to introduce anything that's not already there or hasn't been indigenous, but to see if, slightly as described, can we slightly, this is the whole idea of restoration. Can we restore what's been there previously to benefit the system? And, the whole, and really that's the the whole effort here, and that we know that the system didn't originally have lots of farms and septic systems, didn't originally have a lot of runoff, and possibly originally did have more of an oyster population. And so all of these approaches are really to trying that exact effort to push things back in the direction that they possibly had been.
All right, I don't see any other questions coming up in the chat. If anyone has any they want to actually say out loud, go for it. Otherwise, I'll turn it over to John to say a few words.
All right, there being none. First of all, thank you, Chris, for all the research you've done, and thank everyone for coming. This has been a great turnout and very encouraging in terms of getting the word out to people that live in the watershed as a whole, because I think together as a larger group of people, we can get a lot accomplished, especially with the raising of private funds to leverage public funds as we get into the remediation phase of this work. Chris mentioned, there's going to be continued research that we are going to do to really pinpoint the location of nitrogen-rich groundwater, and then of course analyzing that and understanding if, for example, PRBs are a good solution. That's something that we will be working with Ron on. We will continue to be working with Chris on other aspects of this effort to really inform a management plan that will be commissioned by the trustees. So all of this data will be shared with the trustees as they retain someone to actually prepare a management plan to revitalize the pond. Again, I just want to remind you that we are, as an organization, honored to be playing a coordinating role here. We look forward to working with our public partners to tap the public resources, to supplement the private funds that we raise to do this research. So we will be making an application, for example, in January to the Southampton Town's CPF water quality improvement program to do this work with Cornell to further understand the groundwater and the nitrogen in it and where it's coming into the pond. So we really encourage you to support all of this work by contributing to our Sagg Pond Restoration Fund. And we'll be giving you more information about that in the coming weeks and months as we continue this effort. So it's going to make a difference, all of us working together. And again, thank you so much for being here and participating in this. It's a work in progress, but we've made tremendous strides in understanding what's happening here, thanks to Chris. And we look forward to our continued work together. So everyone have a great holiday. Stay safe. We'll be in touch. Any questions, you can always follow up with us at the Land Trust and with Chris. And we look forward to continuing to see you. We're going to build up the communication with other folks in the watershed. So please let them know what you've learned this evening. This has been recorded. So if you have friends and neighbors that you think should see what was discussed tonight, let us know, and we'll share the link with all of you as well, so that you can pass the word. But I think a great participation, and again, thanks so much for being here. So with that, again, stay safe, and we'll see you again soon in the coming weeks and months.